pleasure to be here, especially the work that Felice did to get us all ensconced and to learn about the polio and stress, which many of the people on horseback must have felt that last few minutes. Like I suspect many others that are speaking today, I have many memories of Wiley as just an incredible person to know. He was really the ideal mentor, as this quote seemed to summarize better than others how important he was to many of us in our lives. Well, the incidence of, I'm going to talk a little bit about obesity today and how it affects one particular aspect of metabolism, which is glucose metabolism. This is an out-of-date slide, but it goes to show that in the United States, at least, the incidence of obesity has really risen in epidemic proportions. And here you can see in 2010, nearly everything is either orange or brown, and denoting that the incidence of a BMI greater than 30 is increasing. Now, the reason this is so important is that it's a precursor for the development of type 2 diabetes. Normally, we keep remarkable energy balance in the calories that we take in and those that we expend. We're about 99.9% efficient. It's just that 0.1% that gets us in trouble. And of course, if you were to go through to Florence today, you might see some of the results. Here, David has not managed to escape that problem. Now, the problem with insulin resistance is that it increases circulating glucose concentrations. And normally, glucose is kept in check by insulin when we feed. The production of glucose by the liver, which happens at night, is downregulated, and the flow of glucose into muscle is increased. And these are two parameters that are specifically affected in individuals with type 2 diabetes. When we fast, we break down glycogen for the first 12 hours or so, primarily, and then that gets replaced by a process known as gluconeogenesis, in which the liver actually makes new glucose to feed the brain and the red blood cell compartment. And it's the activation of gluconeogenesis that really accounts for the chronic hyperglycemia that's associated with type 2 diabetes. Now, so this is part of what I'm going to talk to you about, is the epigenetics of how glucose production is regulated. So by epigenetics, we separate out the effect of the sequences of the DNA, which would be called genetics, from things that happen to the proteins on the DNA, and that's called epigenetics. Many of these proteins, primarily histones, are modified chemically, and I'm going to talk about one modification called acetylation. Okay, so separate from what's happening in the liver, the fat also undergoes low-grade inflammation that contributes to the development of insulin resistance, and this just shows fat cells getting surrounded by macrophages that can release cytokines, and these cytokines in turn contribute to the development of insulin resistance. Okay, now why we think this is important is because the pathway that we study, which is cyclic AMP, turns out to be remarkably immunosuppressive. And in fact, a number of bacteria elicit toxins. They make toxins that actually increase the production of cyclic AMP. So for example, tuberculosis has 15 adenyl cyclase genes that contribute, that allow the bacterium to evade the immune system, and that's why it sticks around so long. Anthrax makes an edema factor. And you've probably all heard of cholera toxin, as well as pertussis toxin, which we use in everyday experiments, but these all go to increase cyclic AMP and downregulate. So I'm going to tell you a little bit about how this second messenger pathway, in addition to its effects in the liver, can also have immunosuppressive effects. So really that's the question we're trying to answer is how does cyclic AMP in one condition act as an accelerator to increase the production of glucose and in another to be a brake, and I misspelled brake here, to decrease the production of cytokines. Okay, so this is just a Ferrari drawing that is sort of giving you a sense of the three molecules, or groups of three molecules that I'm going to be talking about. So we have about 800 or so of these in the genome, and they serve as genetic switches that regulate the expression of genes. You probably all know about transcription factors on the right, the gas pedal that accelerates the expression of the genes of interest. And separate from those are the repressors, which are the brakes, and a group of proteins on the far left, which are coactivators. These really drive up the expression of specific genes and work with the accelerator to increase the expression of programs that are important for glucose and other metabolic events. Okay, so first to the liver. Now, in the fasting state, there are two pathways that are important. One is the downregulation of insulin, so insulin levels go low, and that stimulates a transcription factor called FOXO. In parallel, glucagon increases the expression of the activity of the Krebs pathway, which is the one that I'm going to talk to you about. Okay, so GCG stands for glucagon, and here you can see glucagon attaching to its receptor, activating adenyl cyclase, and then stimulating protein kinase A, which is a heterotetramer. And then the catalytic subunits go into the nucleus and they phosphorylate this transcription factor Krebs, which increases its activity. And as a result of being phosphorylated, it binds to a coactivator. That's the clutch that I showed you earlier. And that stimulates the expression of gluconeogenic genes. Okay, the effect of what I just showed you is not long-lasting, it's transient, and that's true for many stimuli. So here you can see the phosphorylation of Krebs in the center section, and it shows sort of maximal levels of phosphorylation after about 30 minutes, and then after about an hour, even in the continued presence of the hormone, it comes down and it becomes unrecognizable after 4. Okay, so the kinetics of gene induction then are sort of burst attenuation, the burst occurring at about an hour and then attenuating thereafter. We know a lot about the structure of how this happens. Here you can see the phosphate, the phosphorylation site being surrounded by 2 amino acids that coordinate that allow Krebs to bind to CBP. CBP is the coactivator and it's in the background. And as a result of binding, it winds into a helix, which is in yellow, and that stabilizes the complex. Now, separate from this pathway is another pathway that works with the Krebs pathway. These are CRTCs, or cyclic AMP regulated transcriptional coactivators. There's a family of 3 of these proteins. They share a similar structure, and they bind to a different region of the Krebs protein. So here's CBP. In the middle, the one I just talked to you about, and at the end, binding to this B-zip region are the CRTCs. So it's a little complicated, but the bottom line is that they are also regulated by phosphorylation. Under basal conditions, they're maintained in the cytoplasm. In an inactive, they're sequestered, and upon stimulation with glucagon, they become dephosphorylated and enter the nucleus and then bind with CBP to Krebs. So there's these 2 different events, both involving these accelerators and the clutch proteins that I mentioned earlier. This does not happen in isolation. So the genes that regulate gluconeogenesis, and here I've noted glucose 6-phosphatase and PepsiK, they're normally covered and coated by histone proteins, and that prevents their activation. So they need something to convert these into an active state. And that involves acetylation of histones. And upon acetylation, these histones clear out and allow the transcription factors to come in and bind. So I'm just going to show one piece of data that brings home that lesson. This is a chromatin immunoprecipitation assay, and it's looking at the glucose 6-phosphatase gene. And in green would be a cell that's not stimulated, and in blue is what happens when you stimulate the cell with glucagon, or in this case, forskolin. So you can see by the peaks, I'm going to go away from the microphone for just a second. So in response to cyclic AMP, you can see the histones on either side of the Krebs peak becoming much more acetylated. And I won't talk about it, but this protein here turns out to be very critical in setting that up. So in the end, the histones create this little dip here that allows for Krebs to come in, along with the coactivators, CRTC and CBP. What I've just shown you is how Krebs is allowed to bind and then come in and bind with CRTC2. So that allows for the genes to be expressed and for glucose to be made by the liver. Okay, this is important because the process is turned on in diabetes. So here you're looking at normal mice, lean mice, and OB or DB diabetic mice that are overweight and that have high levels of Krebs activity. So this is using an LCD camera to look at Krebs activity in the liver, and the stronger the signal here, the more Krebs is being turned on. And you can see that the diabetic animals have this switch very strongly turned on. We've managed to keep privacy at high levels. For that was a Wiley joke. Actually, I'm borrowing Wiley's. Okay, why this is important. If you knock out the CRTCs, those are the clutch proteins, the genetic switches, now you can see that under high-fat diet or normal chow conditions, the production of glucose through the gluconeogenic pathway is strongly downregulated. And so the genes that are important for that process, glucose 6-phosphatase and PepCK, are also turned down strongly when these coactivators are turned down. And as a result, circulating insulin levels are quite a bit lower, even in the high-fat diet, cafeteria food diet condition. This turns out to be important because these animals that have the knockout of this coactivator, CRTC, are also more insulin sensitive. So if you provide a smaller dose of insulin, it's sufficient to suppress hepatic glucose production. This is work done by Jerry Olewski's lab. And as a result, the amount of fat in the liver goes down. Normally, fat concentrations in the liver go up. It's a condition known as steatosis. And here in the knockout, it's a lot lower. So that suggests that inhibitors of these coactivators might be useful in the treatment of individuals with insulin resistance. Okay. Now I'm going to just move to the second story, having described a switch in which the modification of histones over gluconeogenic genes allows for this program to be expressed. Here I'm going to talk about how cyclic AMP shuts down the expression of cytokine genes that are activated in response to infection or to high-fat diet. So similar to what I showed you before, the transcription factor NF-kappa B is normally sequestered in the cytoplasm by another protein called I-kappa B. And in response to bacterial infection or administration of lipopolysaccharide, this heterodimer is released, partner is released, it goes into the nucleus, binds to this clutch protein, CBP, and which acetylates the histones and allows for CBP to come in and NF-kappa B to turn on these cytokine genes. So if you administer LPS to animals, it's very toxic. The animals die very quickly. But if you co-administer a compound known as roloprem, which is a phosphodiesterase inhibitor that increases cyclic AMP, these animals survive. And they survive because they make a lot lower levels of the cytokines like TNF-alpha and IL-12. So what's the mechanism? Well, it turns out that if you treat with something that stimulates cyclic AMP in macrophages, prostaglandin E2, That prevents the acetylation of histones over cytokine genes. Here I've shown IL-12 beta. So as a result, these genes don't get expressed. So here on the right, this shows a cartoon of what's happening. Normally, in response to LPS treatment, the histones get acetylated and the genes can get turned on. But if you treat with PGE2, the histones don't do that. They stay unacetylated. And as a result, the transcription factors can't come in and stimulate these genes. Here are the proteins that carry out that process. They're histone deacetylases, or HDACs, and they belong to a family, the type 2 HDACs, that are also regulated by moving from the cytoplasm to the nucleus. So under basal conditions, these proteins, here I've shown HDAC4 because that's the most abundant in macrophages. It's highly phosphorylated and it's sequestered in the cytoplasm. Treatment with cyclic AMP agonists like prostaglandin E2 or catecholamines will stimulate the dephosphorylation of HDAC4, allowing it to migrate into the nucleus where it inhibits NF-kappa B activity. So if you look in animals that have a knockout of HDAC4 in macrophages specifically, so that's MKO here, and you stimulate these animals with LPS treatment, you can see, or just even look under basal conditions, you can see that the expression levels of all these cytokines are much higher than in lean controls. And as a result, these animals have more macrophage infiltrates in the fat. Okay. So what's the consequence of doing this? You have more cytokines being produced when you knock out this histone deacetylase. Well, it turns out that if you look under high-fat diet feeding conditions, here on the left, these animals have higher glucose levels, circulating levels during fasting, as well as under ad libitum feeding conditions. That's because they're insulin resistant, and you can see that the levels of circulating insulin are higher in the red bar here. That corresponds to the knockout of HDAC4 in macrophages specifically. And as a result, their free fatty acids are also pretty strongly elevated. So how does that fit into the physiology that I mentioned to you? Well, normally in response to acute overfeeding, leptin is stimulated. Leptin is produced primarily in the fat, and it stimulates the sympathetic nervous system, leading to the production of norepinephrine and the activation of this cascade that promotes HDAC4 localization into the nucleus, where it shuts down NF-kappa B activity. So there is a pathway to shut down cytokine production by macrophages in response to acute overfeeding. The problem is if we overeat too long, this pathway gets downregulated, and so you can't do that anymore. And so these individuals get insulin resistant. Okay, so what I've talked to you about today are two different ways that the cyclic AMP second messenger pathway mediates effects of catecholamines and a host of other hormones. One involving the activation of a histone acetyltransferase in the liver that leads to more glucose production, and one shutting down the production of cytokines by histone deacetylases. These are the people that did the work. I want to specifically recognize Young-Sil Yoon, who did the glucose work, and Bing Luan, who did the work in macrophages. And I want to thank you all for your attention.